This video is to talk you through mechanisms of actions, or modes of action, sometimes called MOAs, of a select handful of different antibacterial, antifungal, and antiviral drugs. Make sure you know the targets of all of the drugs, the structural targets. But within each structural target, there are various ways that you can attack that structure that can disrupt it, that different drugs do in different ways. It's different plans of attack going after the same structure. So that's what we're going to focus on in this video. So the first structure that we're going to look at are a handful of drugs that attack the cell wall. Remember that bacteria have a cell wall made of peptidoglycan, fungi have a cell wall made of chitin, plants have a cell wall made of cellulose. Protozoans and animal cells do not have cell walls. So these are lovely targets for drugs that will not affect humans because we don't have cell walls made of these sugars, or at least have minimal side effects. So a couple of bacterial examples going after peptidoglycan. The bacteria, the antibacterial drugs, the beta-lactam. This is where you find the penicillins and the cephalosporin families. These drugs go after the cross-linking between the peptidoglycan layers. Think of the peptidoglycan layers as kind of like layers of a cake. They all stack on top of each other, and those layers are attached to each other through different bonds. The beta-lactam drugs prevents the attachment of those layers together. So the cake is stacked up but not actually stuck together, so it can easily fall apart. Vancomycin also goes after the bacterial cell wall, but it prevents all horizontal and vertical layers from forming at all. So there's essentially no layer formed at all. The cake doesn't even stick together to make a layer. It's just individual little sugar blocks that don't come together to make the full horizontal peptidoglycan layer. Another group of anti-cell wall drugs are against fungal cell walls. So it's referred to as the chitin cell wall, but it's actually got several different sugars in it. One of the major sugars found in the chitin cell wall is a type of sugar called glucans, a couple of different types of glucans. And there's a group of antifungal drugs called the echinocandins, or you'll hear them called the candins for short. And they're going after that fungal cell wall specifically. Some examples of drugs that go after translation, so going after protein production. Notice our list here, I've given you 4 examples of bacterial translation inhibitors, but no antifungals or any other group. And that's because any organism that's a eukaryote, their ribosomes, their RNA production, polymerases, all of that look like human enzymes. So you can't disrupt those processes without disrupting the human host. So we only have bacterial anti-translation, anti-protein production drugs. So the examples I've given you here are aminoglycosides, tetracycline, chloramphenicol, and macrolides, all going after that ribosome, mRNA, tRNA interaction. But all in slightly different ways. So the aminoglycosides insert themselves into the that mechanism, the RNA plus ribosome mechanism, and what they do is they cause the ribosome to not sit squarely and firmly on the messenger RNA, so it kind of slips and slides forwards and backwards. So what's made is not the proper codon because it's shifting forward and backwards on the RNA. So you get a protein made, but it's not the correct one. Tetracycline actually goes in and essentially competes with the spot where the tRNA would come in to the ribosome. So it blocks the tRNAs from being able to deliver the amino acids to the ribosome, and the whole process stops. Chloramphenicol goes in and disrupts the part of the ribosome where the amino acids would be hooked together to make the growing chain. So it essentially prevents the amino acids from hooking together, so no protein is produced. And then macrolides go on and block the movement of the RNA, mRNA, and ribosome, so that sliding can't happen where the ribosome can move down the mRNA to read it. That movement doesn't happen, so it's basically kind of glued in place. A couple of examples of membrane function drugs. Bacterial polymyxin drugs, they attach themselves to lipopolysaccharide, the LPS, on the outside of gram-negative bacteria. So this is a gram-negative specific drug, and what it does is it basically puts a hole in that membrane. So you've punched a hole in your balloon, and everything will come dumping out. There is a similar drug going against fungal cells called the polyenes, and they attach to a special cholesterol or a special lipid found in the fungal membrane that's the fungal version of cholesterol. It's called ergosterol. And it attaches to that molecule and punches a hole in the membrane. And then there's another very effective group of antifungal drugs called the azoles, and they prevent the production of that ergosterol molecule. So this vital molecule is missing from the fungal membranes. So the polyenes punch a hole to the in the membranes that are already there, and the azoles prevent the production of this vital molecule for future membranes. But both of those, the ergosterol is only found in fungi, so it's specific to fungal cells. And the polymyxins are specific to gram-negative because they only interact with LPS, which is only found in the outer membrane of gram-negative bacteria. Examples of mechanisms of action that are going after a specific metabolic pathway, in other words, a specific chemical reaction. The sulfa drugs are a classic example of that. It's a chemical reaction found only in bacteria that is involved in the production of folic acid. And the drug goes in and interacts with one of the key enzymes in that process and prevents folic acid from being produced. The lack of folic acid doesn't harm humans as much, doesn't harm us directly, because we don't produce our folic acid, we eat it. Actually, we steal a lot of it from the bacteria that live in our guts as well as from our food. Some antiviral examples fall in this category. We learned about HIV pathogenesis, and we learned about AZT, which is a drug that specifically targets reverse transcriptase and blocks that reverse transcriptase step in HIV pathogenesis. And then Relenza and Tamiflu, we also have already been exposed to in the influenza lesson. These are drugs that block the release step, the budding step of influenza virus, so the virus is trapped inside of the cell and doesn't spread. And then the last mechanism of action, the last groups of drugs here that we're going to talk about are drugs that target nucleic acid production. This could be targeting DNA production, like replication. This could be targeting RNA production, like transcription. So for bacteria, we have the quinolone family, which is going after DNA production and RNA production because it prevents the DNA from properly unwinding. When you open up the DNA, it tends to get kind of not gummed up, but kind of knotted up. And there's a special enzyme called topoisomerase that goes in and cuts that enzyme. It goes in and cuts those knots out. So if you block that, then the DNA doesn't open properly and you can't use it properly. And then there's a family of drugs, the rifamycin family, where you find rifampin, that goes after RNA polymerase. So it blocks transcription. And there's another antiviral example in this group, nucleoside, sometimes called nucleotide analogs. These are basically chemicals that look like nucleotides, but they're not, they look like nucleotides for nucleic acid production sense, but they don't actually fully interact like a normal nucleotide would. So what happens when you take one of these nucleotide analogs or nucleotide inhibitors and you stick them into a regular piece of nucleic acid is that nucleic acid is no longer capable of forming all of the correct interactions and structures, and so it's basically kind of garbage. And so when you've got a cell that's been taken over by a virus and it's producing a bunch of virus DNA or RNA, then these nucleotide inhibitors, these drugs, can get in there, and the DNA and RNA that's made at that moment is garbage DNA and RNA. So the virus can't correctly, fully multiply itself to produce effective babies.